Welcome, everyone. Today we're going to break down one of the most elegant and, honestly, one of the most important processes in all of cell biology, apoptosis. This is the body's incredible system for programmed cell death.
Okay, so here's our game plan for today. We're going to start with the absolute basics, what apoptosis actually is. Then we'll get into the nitty-gritty of the different pathways that kick it off, both from inside the cell and from outside signals. After that, we'll connect it to the clinic and see what happens when this whole system goes haywire. And of course, we'll wrap it all up with a nice visual summary to make sure it all sticks.
All right, first things first, let's get a really solid definition down. What exactly is apoptosis? And maybe more importantly, how do we know it when we see it?
So the key takeaway here is that apoptosis is not a chaotic, messy death. It's a deliberate, programmed event that actually requires energy, ATP, to happen. You can think of it like a controlled demolition of a building. The cell uses a special set of enzymes called caspases, which are basically the cell's own demolition crew. They act like molecular scissors, carefully chopping up the cell's components from the inside out in a way that's clean and contained.
Now, to really appreciate how neat and tidy apoptosis is, you have to compare it to its counterpart, necrosis. If apoptosis is that controlled demolition, then necrosis is like an unexpected explosion. With necrosis, the cell literally bursts open, spilling all its guts out, and that causes a huge inflammatory mess for all the neighboring cells. Apoptosis, on the other hand, keeps its membrane intact the whole time. It's all about being a good neighbor, even in death.
So if you were to actually peek at one of these cells under a microscope, what would you see? Well, there are some classic telltale signs. First, the whole cell just kind of shrinks on itself. Then inside the nucleus, the chromatin condenses into these dense little clumps. We call that pypnosis. Next, the nucleus itself starts to break apart into fragments, a process known as karyorrhexis. The cell membrane then starts to bubble out, we call this blubbing, before pinching off into these neat little packages called apoptotic bodies. These are basically bite-sized pieces that are easily gobbled up by scavenger cells. No mess, no fuss.
And you know, on a molecular level, there's another dead giveaway. Those caspases I mentioned, they activate other enzymes that chop up the cell's DNA. But they do it in a very specific way. They cut it neatly between the histones. So if you take that DNA and run it on a gel, you don't get a smear. You get this beautiful, distinct ladder pattern where each rung is a multiple of about 180 base pairs. It's a very clean, very specific signature of apoptosis.
All right, let's move on to our first major pathway. This is the intrinsic pathway, where the cell itself decides, you know what, it's time to go. This is the cell's own command, usually triggered by some kind of internal stress.
It's really a beautiful cascade of events. It starts with a trigger. Maybe the cell got zapped with radiation, or maybe a growth factor it needs to survive, like IL-2, gets taken away. This stress signal wakes up two key proteins, BAX and BAC. These guys get together and literally punch holes in the mitochondrial membrane, and that allows a protein called cytochrome-c to leak out into the cytosol. And once cytochrome-c is out, it's game over. It's the final signal that activates the caspases, and the demolition begins.
Now there are a few key proteins to keep track of here, and this little mnemonic is super helpful, especially for exams. Just remember, BAX and BAC are the bad guys. They're bad for survival because they start apoptosis. On the flip side, you have BCL2, which helps the cell be clever and live. And this really gets at the heart of the decision-making process. BCL2 is the guardian of the mitochondria. Its whole job is to stand in the way of BAX and BAC to keep that mitochondrial membrane sealed shut and keep cytochrome-c locked inside where it belongs. So you can really think of it as a constant tug of war inside the cell. As long as BCL2 is winning, the cell survives. But if BAX and BAC take over, apoptosis is triggered.
Okay, so that was the inside job. But what happens when the order to self-destruct comes from the outside? Well, that's where our next two pathways come into play, and they're often run by the immune system.
The first one is the death receptor pathway. It's pretty direct. You have a signaling molecule, like Fas ligand, floating around. It acts like a key that fits into a specific lock on the cell's surface called the death receptor. A famous one is called Fas, or CD95. When that ligand binds, it's like flipping an external kill switch. That binding event directly sets off the cascade inside the cell.
Now this third pathway is even more direct. It's like a special forces operation. This is how our cytotoxic T cells and NK cells take out threats, like virus-infected cells or tumor cells. The immune cell cozies up to its target and releases these special granules. One protein, perforin, acts like a hole punch, creating a pore in the target cell's membrane. Then another enzyme, granzyme B, slides right through that pore and directly activates the executioner caspases. It's basically the immune system hand-delivering the execution order.
So why are we spending all this time on these molecular details? Because as future clinicians, you absolutely have to understand that when this beautifully balanced process of apoptosis goes wrong, it leads directly to some very serious diseases.
And this table shows you exactly what I mean. Take a look at follicular lymphoma. There's a specific genetic screw-up, a translocation, that causes the cancer cells to make way too much of that anti-apoptotic protein, BCL2. So these malignant cells become immortal. They simply refuse to die when they're supposed to. That's a disease of too little apoptosis. The same goes for a condition called ALPS. Here, the problem is a faulty FAS-L pathway. The body can't get rid of immune cells that attack itself, which leads to autoimmunity. Again, a problem of too little apoptosis.
Okay, let's take a deep breath and pull everything we've just talked about together into one final, clean summary. This will really help lock in the concepts.
So here it is, all on one slide. We've got our three main pathways. The intrinsic pathway, triggered by internal stress. The extrinsic pathway, triggered by an external death signal. And the perforin/granzyme pathway, which is a direct attack from an immune cell. But look at the last column. This is the big takeaway. Despite their different triggers and their different key players, all roads lead to the same place. They all converge on one single critical event, the activation of the caspases. That is the point of no return.
So I want to leave you with this final thought. We've seen that apoptosis is all about maintaining this incredible balance between order and chaos, between cell survival and cell death. As you go forward in your studies, I want you to look for this theme, because you're going to find that this concept of maintaining a delicate balance is at the absolute core of health and disease. Thanks for tuning in today.